So if it's okay, I thought we might wait just a couple minutes more to give any latecomers a chance to log on. But I can start with the introductions. As many of you know, Heritage University is conducting a search for a professor of biology. And Dr. Sund made the recommendation that this be a, a directed search, meaning that there's a specific candidate in mind that we were very interested in, and that candidate is Corbin Schuster. So we're all extremely pleased that he was happy to apply for the position. This is one of two talks that he'll be giving as part of the application process. This is a teaching talk. I've instructed Corbin to give a talk that's approximately 40 minutes long, and that is on a topic of his choice, but in the subject of microbiology. The faculty position is for an assistant professor of biology with a specialty of microbiology. And that, of course, is what Corbin is specializing in. So briefly from his bio, he is from our area, and he is a Heritage University grad. He went to Zillow High School, and then he graduated from Heritage in 2018 with a BS in biomedical science. He then went to Oregon State University, and he is one of those remarkable students who went straight to a PhD program and is expected to complete his PhD in a very short amount of time. So we're very excited to hear that, and really, very impressed how quickly he's done that. Corbin is a enrolled member of the Academy Nation, and he was very active while on our campus. And based on Facebook posts, he's very active on the Oregon State campus. And I am thrilled to have a candidate with so much energy, positive energy, and just who has applied. And so we're very happy to have you, Corbin. And this is the first of two talks. The second one will be on Friday at noontime as well. And I will be recording these and posting them. I haven't figured out where, but I'll post them. I'll probably put them on YouTube and then share the link for those who would be interested if you want to review them. I am recording this again, just so everybody is aware. Okay, so I think with that, Corbin, I think this is a teaching talk, so I'm actually going to let you direct how you want to handle questions during, questions at the end. So I'm going to let you decide that for this one, and you feel free to direct us.
Perfect. Perfect.
But take it away.
Oh, sorry. Thank you so much for that introduction, Jessica. Shek or hello everyone. I'm going to be giving a microbiology 101 lecture. Jessica had directed me to make sure that it was a 101 lecture, and I felt pretty confident with going forward discussing cell structure and function. And I figured today we'll talk about bacteria, eukarya, and we'll compare and contrast the two. And if we have time, we'll go over archaea as well. To orient us, I suppose, since we're on Zoom and not necessarily in person, I really want to utilize the chat feature. And so if you have a question throughout the duration of the presentation, I will monitor the chat. And so I'll use that as like the raise hand function. So if you have a question, definitely use the chat, and I will try to integrate the chat throughout the duration of the lecture. So before we get started, I want to orient ourselves as to I want to orient ourselves as to our current knowledge with the with the structure and function of a cell. And so using the chat feature, what are some common components of a cell? And like using the chat feature, go ahead and just dump up any answer that you have in the chat. Awesome, awesome. Yes, these are all perfect. Yes, these are all important components of the cell. We have both external structures of a cell and internal structures for the cell. But for now, I want us to orient ourselves and let's focus on the cell membrane, the cytoplasm, and the nucleus. And for now, think of the nucleus as the center or the core of the cell's DNA. So our learning objectives for today is we're going to go over the general life processes. We're going to compare and contrast prokaryotic versus eukaryotic cells. And we're going to look at the cell structure and function of these different life forms of bacteria and eukarya. But before we jump into the nuts and crannies of cell structure and function, we need to understand what we define as life as biologists. And so there's 4 general characteristics of life. There's growth, reproduction, responsiveness, and metabolism. Growth refers to an organism's ability to increase in size. That means for humans, for example, we're able to grow from infancy into adolescence into adulthood. Whereas reproduction refers to an organism's ability to reproduce. And this occurs either asexually or sexually with gametes or sex cells. But responsiveness refers to an organism's ability to respond to their environment. For us as humans, our ability to respond, oh, it's too hot, I'm going to move into the shade. Oh, it's too cold, I'm going to move into the cold. But there's much more complex networks here, is because we also see responsiveness in the genetic realm, and we call this epigenetics. And so these environmental factors impact our genome and how we are able to sort of live within our environments. And metabolism refers to an organism's ability to consume nutrients foreign to the organism, so bringing in nutrients from outside for biochemical breakdown into energy sources. But before we move on, we also have to understand what does not fit this description of life. And as we are all very familiar with, SARS-CoV-2, the novel SARS-CoV-2 virus, which causes COVID-19, does not fit this description of life. And there's several reasons why. It doesn't have cells, and there's no cytoplasm or cellular organelles within. And so that means that it does not necessarily carry out its own metabolism. It actually will hijack a host cell a host cell metabolic machinery, right? And so although it does not a form of life, it does infect all forms of life. And and so I just wanted to make that clear, that we're going to talk about specifically today, not viruses, but bacteria and eukarya. So we need to understand the evolutionary relationship too with these different bacteria. And so this Hug et al. Paper from 2016 describes the domains of life. And these domains of life, this phylogenetic tree essentially tells us the different phylogenetic relationships with these different life forms. And so bacteria, which up here at the top, are separate or sort of more evolutionary distinct than what we see with archaea and eukarya. Archaea clusters with eukarya, even though the structure and function more resembles bacteria. So it's a very cool evolutionary phylogenetic relationship here. And the focus on this isn't necessarily that we need to understand and memorize all of these different groups and clades. It's more that we want to understand the evolutionary relationship between these different life forms. And so moving forward, cells are living entities. So as we know, life is composed of cells. That means that organisms need to be are capable of all these different characteristics of life that we described. And with that, there's different cell types. So there's single-celled organisms, multicellular organisms, which are much more complex, like eukarya. And all cells can generally be described as either prokaryotic or eukaryotic. Sorry, I thought there was a question in the chat. And so moving forward, when we talk about this, scientists often they'll categorize organisms based on shared characteristics into groups called taxa. The prokaryotes is characterized oftentimes when bacteria and archaea are being talked about, but prokaryotes typically lack membrane-bound organelles. And by that, I mean organelles within the internal structures of this, such as this nucleoid, the DNA for this prokaryotic cell, it is not bound within a membrane that protects it and segregates it from the rest of the cell. Whereas in a eukaryotic cell, such as the nucleus, it is bound within this nuclear envelope, which protects it and segregates it from the rest of the internal structures of the cell. Although archaea cluster evolutionarily to eukarya, they are unicellular organisms. So they don't necessarily have a nucleus, and therefore, like I say, their structure and function more resembles and is more characteristics of prokaryotic. And so this is just to orient us as is that the word prokaryotic comes from the Greek phrase meaning before nucleus. And so here, and we see the evolutionary relationship. So before nucleus and true nucleus. And so although there are many different cell types, all cells share general characteristics of life. And today we'll discuss exactly how biologists and scientists in general are able to distinguish between bacterial, archaeal, and eukaryotic characteristics, and how those characteristics I work with diseases a lot. So in how those characteristics may influence a pathogen's ability to actually infect its host. Yes. How does this relate to the human body? Great question. This certainly relates to the human body is, we're made up of eukaryotic cells, right? So we're eukaryotic organisms. And how this relates to the human body is that our entire bodies are made up of different cell types, such as we have different blood types, different blood cells, as well as different immunological cells, which play essential roles in making sure that we're maintaining our health status and moving forward in a good way. But before we get into, I guess, the nuts and crannies of the functions of these different components of a cell, we need to understand what the external structures of the bacterial cells are, influence a pathogen's ability to persist. And so one of the external structures of the bacterial cell is the glycocalyx. And so some cells have this gelatinous sticky substance that surrounds the outside of the cell. And this is called, the glycocalyx, but it plays a critical role. So let me just step back. It plays, this is the glycocalyx, and it is this layer that surrounds the entire cell, and is composed of polysaccharides, polypeptides, sometimes even both, but they are produced inside the cell, these molecules, and then they're pushed out to extrude into the cell surface to cover the entire cell surface. So these glycolyses, they protect from desiccation, which is essentially drying out of the cells, and they can play a major role in a pathogen's ability to survive and cause disease, as they provide this sort of fluid structure around this slime layer, essentially. Interestingly, biofilms are colonies of bacteria that conjugate and influence their persistence on a surface. This is huge in the medical field, such as surgical equipment. We see biofilms will colonize an entire surface of equipment and commonly used equipment. And this may cause, this may be a concern for pathogens such as Microbacterium. There's also a flagella. So how do the cells move around, right? And so within their environment, the flagella, these are long structures that will extend beyond the surface of the cell, propelling the cell through the environment. Using video enhanced light microscopy, we're actually able to visualize this. Regular light microscopy doesn't scatter the light good enough to see this, so we have to use these special sort of techniques. But using video enhanced light microscopy, we're able to see these different flagellar types. And so they have different arrangements. I believe this is an E. coli. And what's characteristic of the E. coli is that these flagella types actually have this flagella sort of coming out, not randomly, but sort of all over the cell surface of the entire cell, right? Whereas some species will have this more amphiphychicus representation, where they have these flagella types coming out of they're not polar, but the polar ends of the bacteria. Whereas in some species, such as the Vibrio species, we're going to only have a single flagella come out. The bacterial flagella are typically composed of 3 parts: the long, hollow filament, a hook, and a basal body. And so here if you will imagine that the cell, is, extending beyond here, but you have your outer membrane, and then you have this this filament that extends into the environment and is composed of identical globular molecules called flagellin, which is this flagellin. And this hook is characteristic of prokaryotic bacteria. And it sort of sits there. I'm going to use my arm as reference. Sort of sits there, and it'll tumble, and it helps the bacteria to tumble and run. And so here, I actually have a picture of that. And so as you see, it sort of uses that tumble, and it sort of runs and tumbles. But the rotation can be reversible, but for the most part, they move in response to stimuli. So the bacteria is going to move, like, let's say, for example, that this environment, this cellular environment here is pretty toxic, and it's not favorable for the bacteria. So it's going to want to move out of that. And so it's going to signal, and it's going to move towards a chemotaxis, towards an environment that is actually favorable for the bacteria. Phototaxis is a response to light. And so it's going to move away or towards light. But what's really cool is when we get into the nuts and crannies of the different arrangements, is we see like they're pretty specific to different species of bacteria. And so we have this monotrichous, flagella type that's common to the Vibrio species. And this is one single flagella that comes out. And the Lophotrichus, which is common to the Bartonella species, like I mentioned earlier, it has just one multiple flagella coming out of one end of the actual bacteria. And then Amphitrichus is as common in Spirillum species, where we see the multiple flagella coming out of both ends of the bacteria species, what we see here. And then common bacteria is E. coli, which I mentioned earlier is Peritrichus. It covers the entire cell. Oh no, typo. I'm sorry. That's supposed to be fimbriae. But the fimbriae are sticky bristle-like projection used for adhesion to one another and structures in the environment. There's usually hundreds per cell. They're much smaller than what we know with flagella, but they're used in adhesion to other bacteria and structures in the environment. And so this organism here is Necessary gonorrhoeae, and this is the gram-positive bacteria that causes gonorrhea. This gonorrhea colonizes the mucous membrane of the reproductive tracts in humans, but where the fimbriae come in is to adhere to the host cells within the reproductive tract. And so these tiny bristle-like projections here will actually adhere to different host cells. And these are also common in the formation of the biofilms that I mentioned earlier that colonize surface areas. Interestingly, there's a lot of research going on right now over quorum sensing and the ability for these bacteria to conjugate together and to actually communicate with each other and have these very intricate networks of communication with each other. Interestingly, too, there's also papers that suggest that viruses are able to hijack this communication network and use their communication network for the virus's ability to persist. excuse me, a special type of fimbriate is pili, but they're much longer. There's usually only one to a few present per cell, and these cells use pili to exchange DNA via conjugation. And this conjugation is a temporary union between two bacterial cells or multiple cells for exchange of genetic material. I'm not going to get too far into conjugation because this is a whole topic on genetic microbial genetics, but I did want to bring out that this is an important external structure. So now we get into the important part. Well, I mean, they're all important, but these bacterial cell walls. And so these cell walls are, with bacteria specifically, are how we're able to differentiate between two different bacteria types. So the bacterial cell walls provide structure, shape, and protection, overall protection, and they're composed of peptidoglycan. The peptidoglycan is a complex polysaccharide composed of alternating sugars, this N-acetylglucosamine and the N-acetylmuramic acid, NAG and NAM. Is so going forward, I'm just going to call this NAG and NAM, if that's okay. Sorry, we had a question. Yes, there are a virus can adhere to bacteria as well as other non-bacterial cells. The virus there's actually viruses that infect bacteria as well. Okay. So the peptidoglycan is chains of NAG and NAM and are attached to, I'm sorry, I should have, I guess, read out this question. So there was this question of, so a virus can adhere to bacteria as well as other non-bacterial cells. And that was where my response came in, that yes, there's viruses that can adhere to bacteria and actually infect bacteria. Going back to the lecture, scientists are able to differentiate between bacteria based on their cells using a gram stain. So using using the same gram stain, we're able to differentiate between gram-positive and gram-negative bacteria. And this is based off of their the actual makeup of the bacterial cell wall. And we'll jump into that. So we need to understand the peptidoglycan structure. So this is a layer, the protective layer of polysaccharides made up of the NAG and NAM subunits. And the chains are cross-linked to one another through this tetrapeptide. And so you have alternating sugars that extend off of the NAM sugar. I'm sorry, you have the tetrapeptide that extends off of the NAM sugar that allows this lattice-like structure to form. And I know this is like, but the important part to mention is the structure here and how these are sort of put together. So the 4 amino acids that compose the tetrapeptide are L-alanine, L-lysine, and D-alanine. So this is where I wanted to get into, is the gram-positive bacterial cell wall. And so these cell walls of gram-positive bacteria are composed predominantly of this peptidoglycan structure here. It's almost 90% of the entire cell wall, with layer after layer forming around the cell membrane here, which is composed, we'll get into that, but that's composed of the phospholipid bilayer. The additional component to this, other than the peptidoglycan, is the teichoic acid, which is a glycopolymer which is embedded within these peptidoglycan layers here. The teichoic acid, there's a lot of studies going on, but it is believed to play several important roles for the cell, such as generation of a net charge of a cell, which is essential for development of the proton motive force. But the teichoic acid also contributes to the overall rigidity, right, of the cell wall. So it's important for the maintenance of the cell shape, and particularly for like rod-shaped organisms. So what's important to stay here is that, so the gram stain is essentially these heavy purple stains, and these purple stains will penetrate through this layer and be able to actually stain these different bacterial cells to this color. And we deem this as gram-positive bacteria in the fact that their peptidoglycan layer is their protective layer essentially. This bacteria is Ranibacterium salmonarium, which is the most prevalent pathogen found in migrating salmon in the Columbia River, and it can cause up to 80% mortality in these fish species. It causes what's known as bacterial kidney disease, and so it's an important pathogen of the region. Gram-negative bacteria are a much more complex structure, as we will see. This you have your cell membrane, which is your phospholipid bilayer. You have your peptidoglycan layer, which we just discussed, and then you also have this outer membrane, which we'll get into. But the peptidoglycan layer only represents, in this case, in gram-negative bacteria, about 5 to 10% of the total protection that we see there. The outer membrane is composed of this lipid bilayer, which is very similar in composition to the cell membrane, in that it has the polar heads, the fatty acids, and the integral proteins for transport. So these proteins allow the channeling through these membranes. But how this differs from gram-positive bacteria is this presence of large molecules known as lipopolysaccharides, which are anchored to the outer membrane here and project into the cell environment. The LPS is made up of 3 different components. It's made up of this polysaccharide here. It's made up of the O polysaccharide, the core polysaccharide, and the lipid A. The lipid A anchors the polysaccharide to the membrane. But what's important here is that the LPS is known to serve many functions, including the net negative charge for the cell, essentially helping to stabilize the outer membrane and provides protection for chemical substances to enter the cell. So LPS, interestingly, in regards to pathogens, also plays an important role in the host response to pathogenic gram-negative bacteria. So our body systems, such as our immune surveillance, will actually recognize this LPS, and it will attack it. And it'll attack it until it'll try its best to attack it. And so just like the COVID-19 virus or vaccines, our bodies will build antibodies against specifically the O polysaccharide, actually. And it'll try to break down that gram-negative bacteria. And because of the structure of this, we have the outer membrane, we have the peptidoglycan layer, and plus we have this cell membrane. So you can imagine that purple stain isn't going to penetrate as hard and rigidly as we see with the gram-positive bacteria. And it actually stains the bacteria this pink color. And this bacteria is Salmonella, which is a common foodborne pathogen. So there's also bacteria with no cell walls. And so they lack a cell wall altogether. And this phylum, the general phylum that don't have cell walls is Teneracutes. They're extremely susceptible to osmotic changes, and they use sterols to actually strengthen the cell membrane. This is a characteristic common in eukaryotic cells, because this is unique. So, but what's hard about this is that they're cultured organisms, right? So they're hard to characterize with traditional microscopy. And so we use genomic techniques to characterize and differentiate between them and to understand their evolutionary relationship. And so phylogenetics, in this paper, they were looking at these different uncultured environmental Teneracutes that provide insights to novel ability for pathogenicity. So jumping inwards, so we moved in from the peptidoglycan layer, and now we're going to talk about the bacterial cytoplasmic membrane, which I was talking about with the phospholipid bilayer. So this phospholipid bilayer is bipolar, and it contains these phospholipid molecules, which have different charges at the different ends. So the phosphate containing heads of each phospholipid molecule are hydrophilic, whereas the tails, which sit on the internal side of the bilayer, are hydrophilic or I mean, sorry, hydrophobic. And, and together the tails of in the interior of the membrane, they stay away from the water, essentially. So about half of the bacterial cytoplasmic membrane is composed of these different integral proteins inserted amongst the phospholipid. These integral proteins are essential for channel transport of molecules across this membrane. The peripheral proteins are loosely attached and to the inner, if you will, imagine this is the cell. And then if these are the peripheral proteins and they're loosely attached to the inner and outer surface of the phospholipid bilayer, and they also perform, or their function is in transport and signaling. Now, the fluid mosaic model helps us to describe our current understanding of this entire structure, this membrane structure. We know that the fluid, the proteins and lipids are free to flow laterally within a membrane, and that the membrane proteins are arranged in tiles like a mosaic, like an art piece here. But what I didn't cover there was the cholesterol. So the cholesterol helps to restrict the passage of molecules by increasing the packaging of phospholipids. And so here we can see the cholesterol, it actually helps to package those phospholipid molecules and to make them much more tighter and compact. The carbohydrates, I usually provide these tables for students just as quick references, but I'm not going to go over all of this. So the carbohydrates is used in cell recognition and adhesion. So to summarize, the function of the cytoplasmic membrane, it provides a protective barrier, which allows, which keeps toxins out. It provides a structural support and separation of contents. It provides a selective permeable membrane, so that it allows only what it wants in, and it keeps anything toxic out. These proteins facilitate the transport through these channels, and it helps maintain a concentration and electrical gradient. So passive processes. So let's talk about transport into the cell now. And so these passive processes. I'm sorry. There's two processes here. There's a passive process, which does not require any energy for molecules to be moved into the cell. And then there's active processes, which we do require energy to be moved into the cell. And so the passive processes that we know is diffusion, facilitated diffusion, and osmosis. Direction of the movement usually occurs from a low concentration into a high concentration. And diffusion is the net movement of a chemical down its concentration gradient through the phospholipid bilayer here. Only chemicals that are small and lipid soluble will, through this route, at least through diffusion, will diffuse through the cytoplasmic membrane. And these are molecules like oxygen, carbon dioxide, alcohols, but molecules that won't fit and will not do that through diffusion is glucose, bigger molecules like glucose and proteins. In some cases, proteins can even act as channels or carriers to allow these certain molecules to diffuse down the concentration gradient. And that's where facilitated diffusion comes in, where we have a non-specific protein channel where it'll move non-specific molecules through. And then we also have facilitated diffusion where it is specific in that the binding of the substrate causes a shape change to the channel protein. And these are only when specific, excuse me, my goodness, specific molecules make it through this membrane. Osmosis, of course, refers to water. So osmosis is the diffusion of water across a semi-permeable membrane, which is soluble only to water, not other solutes. So even though there's proteins and amino acids and everything else probably out here, this channel is only going to relate to water. Water molecules can cross from the side of the membrane that contain a higher concentration to the side with the lower concentration, and water will cross the membrane until equilibrium is achieved, essentially. And we're going to jump into that. And so we'll understand the importance of chush, which is water. Chush iwawa kishwit, life is water, or water is life. And as we know, all life forms revolve around water. And so I have this video here. I'll provide a link in the chat in case anyone wants to look at this later. Oh, I think that went to direct message. Sorry, everyone. But we're not going to watch this entire video. I just want to highlight the osmosis portion.
Water passes through the phospholipid of water molecules across the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
Yes, so we commonly compare solutions according to their concentration of solutes, right? So when solutions on either side of a selectively permeable membrane have the same concentration of solutes, the two solutions are said to be isotonic. That is to say that the cells in the isotonic solution experience no net movement of water across the membrane, whereas cells in a hypertonic solution shrink because of the net movement out of the cell, whereas hypertonic, like it was described, hypertonic solutions undergo a net gain of water, and oftentimes animal cells often burst because they lack the cell membrane, or the cell wall. So now we get into the active transport. And active transport requires energy, it requires ATP. And so like facilitated diffusion, active transport utilizes transmembrane permease proteins. However, the function of the active transport proteins requires the cell to expand ATP to transport molecules across the, or not expand ATP across the membrane. And so here it uses the ATP and it moves molecules through this gated or controlled channels. Group translocation occurs in only some bacteria, and these molecules are chemically changed during the transport. These are often molecules such as glucose, mannose, and fructose, different sugars. And so this is a summary figure for the different passive or transport processes. So once we get into the internal structures, we start to understand, or we start to look at the actual cytosol. And so the cytosol is the liquid portion of the cytoplasm. Recall the hallmark of prokaryotes is the lack of a phospholipid membrane encasing and surrounding its DNA. And so here you have the nucleoid all wound up, and that sits in the cytosol, the liquid portion of the cytoplasm. And that's the mostly made of water, but contains ions, carbohydrates, proteins, lipids, and waste. The cytosol is the cytosol is the site of some chemical reactions, namely the production of amino acids and the degradation of sugars. Inclusions and non-membrane organelles. These the inclusions are deposits, energy reserves, and energy reserves. And so these are often carbon and energy molecules like glycogen, and then a lipid polymer, polyhydroxybutyrate, PHB. But what I really want to highlight here is the non-membrane bound organelles of ribosomes and the cytoskeleton. All ribosomes are composed of two subunits, a polypeptide and a ribosomal RNA. But the cytoskeleton is essentially, cells have these external scaffolding and these fibers that allow the cell to essentially wrap around a cell and divide it into two cells. In this photo, we're showing the structure, but we're also showing the the fibers can actually segregate areas of the cell. And that's sort of, it's very hard to see, but that's what they're showing here. There's also spore forming bacteria, and I'm not gonna require everyone to memorize the cycle, but it's important to recognize that when conditions are not favorable for a bacteria, the endospore. And what the endospore does is it allows the bacteria to persist in the environment, and is extremely resistant to radiation, heat, toxic chemicals. And so it leaves this vegetative state, this general cycle, and it turns into this spore, to where the spore protects it, and it has this multi-layered coat. Essentially, it contains that contains the DNA and protects that. Once the conditions are favorable, it moves into back into the vegetative state. The important bacteria to know in this case is the Bacillus. Bacillus species and the Clostridium species. Bacillus species is a gram-positive, or they're usually gram-positive bacteria, and rod-shaped bacteria, and they're non-pathogenic. In fact, Bacillus subtilis can be found in our gut microbiome as well as in soils. Clostridium now is a pathogenic gram-positive bacteria, and this has caused botulism in humans. So now we're going to sort of transition. I think I'm running out of time actually, but we're going to transition to the external structures of eukaryotic cells. And we're going to transition over to eukarya real quick. And so the internal structures of the eukaryotic cells is they too have the glycocalyx, but they're largely absent in cell walls, or with eukaryotes with cell walls such as plants and fungi. These anchor animal cells to each other, and they strengthen the cell surface, while they also provide protection against dehydration, just like in prokaryotes. The eukaryotic cells and the cytoplasmic membranes, all eukaryotic cell membrane, the eukaryotic membranes differ from bacterial membranes in several ways. The eukaryotic membrane contains steroid lipids, sterols, such as cholesterol in animal cells, that will help maintain the membrane fluidity. And at high temperatures, these sterols will actually stabilize a phospholipid bilayer by making it less fluid. That's the opposite with low temp sterols, right? And so these are typically composed of various polysaccharides, and these in plant cells, they actually use cellulose. So, but they contain regions of lipids and proteins and membrane rafts, essentially membrane rafts. Well, similar to prokaryotes, they control movement in and out of the cell. The functions is that they're selectively permeable, and these proteins act as porous channels and carriers to facilitate transport. So in this case, the molecules are brought into the cell through endocytosis, which if a solid's brought into the cell, that's called phagocytosis. And then if a liquid's brought into the cell, we call this pinocytosis. And so we see that these different bacteria will, sort of take up these molecules, and they'll phagosome them into this food vacuole, which is essentially broken down by lysosomes and peroxisomes. So the structure and arrangement of eukaryotic flagella, they differ structurally and functionally from the prokaryotic. Like if you can recall, the prokaryote had that right angle hook, whereas these ones don't necessarily have that right angle hook. The flagella is contained within the cytoplasmic membrane, and the shaft is composed of these tubulin arranged that form these microtubules. And so the flagella have no hook, but the filaments do anchor this basal body here. And so what we're showing here is this basal body, and this sort of extends in from the centrosome. We'll get into that in just a sec, but it has this 9-to-2 arrangement. So you can see it has like these nine paired microtubules with the central microtubule as the 9-2 arrangement. This is very characteristic of eukaryotics, and their basal body is a 9-0 arrangement. They don't tumble and run like bacteria do. Rather, they undulate rhythmically to sort of push and pull the cell through the environment. And so the eukaryotic flagella in the cilia are what they use to for their motility. And so in this case, this is Giardia lambria. I don't know if anyone's ever heard of beaver fever, but this is common in our river systems. It causes extreme diarrhea in humans. Anyways, it uses these different flagella types to move within the environment, both outside of the host and inside the host. This one is paramecium, and it uses these cilia to sort of, it's weird, but they sort of move around, and that's how it's sort of able to be motile. The paramecium is a eukaryotic unicellular cellulite, which is it's found in, like ponds, river systems, slow moving waters. Eukaryotic membranes. So these, just like the prokaryotes, they have non-membrane-bound organelles, and these are ribosomes and cytoskeletons, and they have a similar function, but they're much larger than what we see with prokaryotic cells. And with the cytoskeleton, they have a more complex network of fibers. So this is the non-membranous or non-membrane-bound organelles consist of the centrosome, centriole, and the ribosomes. The centrioles and centrosome animals and some fungi cells contain two centrioles, but what I want to point out here is that these the centrioles are found within the centrosome, right? And so the centrosome is by the nucleus, and they form at 90-degree angles. And with this, what's critical to the centrioles is they're critical in mitosis, but they're also critical in the formation of flagella, that basal body. If you recall that 9-0 arrangement, that's what we see here with these centrioles. And so this is essentially the start of that flagella type. Now, the membrane-bound organelles are what make eukaryotes special. Is the peroxisomes, the Golgi body, secretory vesicles, mitochondria, and the rough ER, the endoplasmic reticulum, as well as the nucleus. These organelles are surrounded by a phospholipid bilayer, which segregate them from the rest of the internal structures. The nucleus is spherical, usually the largest organelle in the cell. Some cell types have a single nucleus, whereas others have multiple, so they call that multinucleate. This is essentially the control center of the cell. It contains most, not all, but most of the genetic information for the bacteria. And in fact, some cells, such as mammalian red blood cells, they will lose their nuclei and only survive for up to a few months. And so the semi-matrix of the nucleus is called the nucleoplasm. And within the nucleoplasm, there are specialized regions where RNA synthesis takes place, which isn't labeled here, unfortunately, but they're called the nucleoli. And so that's where the RNA synthesis takes place. The nucleoplasm, importantly, contains this chromatin, which are thread-like masses of DNA associated with histones that play a critical role in packaging DNA. If you recall from general bio, during mitosis, chromatin becomes visible as chromosomes. And then surrounding the entire nucleus is that envelope, the nuclear envelope, the plasma membrane, which is composed of two phospholipid bilayers. The endoplasmic reticulum, the endoplasmic reticulum is for transport of proteins throughout the cell. And so the rough ER specifically is for proteins, whereas the smooth ER synthesizes lipids and moves that throughout the cell. And so this is the smooth endoplasmic reticulum here, and this is the rough ER. Proteins produced by the ribosomes on the rough ER are inserted into the central canal and of the rough ER and transported throughout the cell. And so the endoplasmic reticulum transport. The Golgi body are flattened hollow sacs surrounded by phospholipid bilayers, and they're shipping for, this is essentially the shipping and receiving, right? In that it receives, processes, and packages large molecules for export from the cell. The lysosomes, which we talked about during phagocytosis, are extremely important. They function to store and transfer chemicals within cells, and within animal cells, they contain catabolic enzymes that damage. And I know this is blurry, I'm sorry, I really wanted this image and I couldn't get a good one of it. But essentially what this is describing is if you have a mammalian cell, for example, and a bacteria infects a human, for example, this bacteria is sort of invaginated and brought into the cell so that it can be broken down and attacked with these lysosomes. And once it does that, it excretes the material through exocytosis. The mitochondria is the powerhouse of the cell. And I don't mean to be taking up so much time, but the two membrane composed of phospholipid bilayer, and this produces most of the cell's ATP. It's an elongated structure found in most eukaryotic cells. And like the nuclei, the mitochondria has two phospholipid bilayers. The inner membrane, though, is forms of these numerous folds called cristae that increase the membrane surface area. And this cristae will produce the ATP in the eukaryotic cell. So you can imagine why we want an increased surface area here. And this is my last slide, the endosymbiotic theory, which essentially talks about the eukaryotes form the union from small aerobic prokaryotes with larger anaerobic prokaryotes. And they essentially what happens was that a cell took in this aerobic bacterium, and this is what we consider, and they formed into membrane-bound organelles. And so this is what we know as the ancestral eukaryotic, where it took in all these different aerobic and anaerobic parasites or bacteria, and eventually lost the ability to exist independently, but retain the portion of their DNA. Okay.
Great. Thank you very much, Corbin. Yay.
Good. Thank you. I'm sorry, I went a bit there. I did have a question. As a student, why is this important to me as I move forward into the biomed pathway? That is a very good question. This is really the basis of the building blocks for understanding pathogens and how they're able to sort of persist, right? So for me, well, I say pathogens because that's sort of where my expertise comes from, I suppose, but it's really core to understanding how organisms come about and how their physiology functions. And so understanding the very basic unit of life as a cell is crucial to understanding the complexity of complex organisms such as humans.
It's pretty amazing. As a literature major, I'm just going, wow, but all those diagrams that kept me engaged. And I was saying, well, there's so much I don't know about. So thank you.
Thank you, Mary. Thank you so much.
Oh, are there any more questions that any of you are wondering about?
If I could make a comment, I really appreciate, Corbin, you bringing in the Chush and that indigenous piece into your lecture and applying it to what you're trying to teach the students. Yeah.
Certainly. Thank you, Maxine.
Just to follow on from Maxine, you had some references to our various water systems and to the salmon, which was also something that was grounding.
I thought it was fun when you were talking about the rotation of the flagella and you put in beep beep, and you talked about shipping and receiving. And I think those things intermixed with your lecture, with your teaching in the classroom are going to wake students up, and they're going to say, okay, this is really fun, and I need to really pay attention because this has an application to my experience, my human experience. So I thought that was really great. And I had a quick question, Corbin, about, I love the mosaic model. I thought that was really effective and beautiful, and it really talked about the pathways that they were trying to describe. And I'm wondering, and the Chush that Maxine mentioned as well, I'm wondering if you see connections as you go along, projecting, you'll see connections as you go along to have a model, a cultural model that will describe more of those aspects of the cells that you are talking about so passionately.
Yeah, certainly. I know there's I'm sorry, there's sort of blank in my head, but I've written papers on just connections with our origin stories, as you know, with the Yakama people and with these different molecular biology theories. And so I think it's important for me, you know, as a instructor, is to sort of integrate these and sort of bring about this knowledge, because oftentimes we see that indigenous knowledge is not brought into the Western philosophy of what we call teaching. And so I think bringing in this in and just, you know, including everyone in all these different knowledge systems into that is crucial.
Thank you. That has profound reverberations. It's really wonderful.
Certainly, thank you.
Just as a neurodivergent person, like, I appreciated your tone, because your tone kind of changed, and that kept my brain engaged. Like, I very often am like, oh, shiny thing, you know, just get distracted very easily. And your different tools and pictures and just kind of the general structure of it, I really appreciate it, because I was actually able to concentrate, which is something that is very hard for me to do. So I applaud you with that, and I really appreciate that.
Thank you.
I just wanted to say I appreciate your passion. I could tell you were like really into this, and I was like, oh my God, he loves this stuff, like trying to give it to us. And that was really, really good. I totally appreciate your passion. And it just like started making me think, oh, really? But then you moved on to some of, oh, really? Oh, it was really good. So thank you for your passion.
Absolutely. Thank you.
Corbin, you took me back as a learner. I was like, oh my God, I thought I'd put this aside, but it all came back. And so I appreciate that. And, speaking of aerobic and anaerobic bacteria and, all the mitochondria, and it was like, oh my gosh, I've been there, done that. Thank you.
Certainly.
I flunked by a 101. I feel like I could pass it now. No, I'm just kidding. I'm joking. I didn't flunk by a 101.
But as I put myself in this place of the student, and, as you were delivering your lecture, I was trying to, get into that space of, the delivery and content and making those connections. What really stood out for me is when you, as a native, if I were a native student, making some connections that are relevant to my place of space. Yeah.
Absolutely.
Great. Well, it sounds like we're wrapping up with our questions. And we're about to hit 1 o'clock, where I know it's like the witching hour for a lot of us. We got to disappear.
No worries. That sounds great.
And I do want to encourage you. So this was specifically a teaching lecture. His research lecture will be on Friday, where Corbin will talk about all the things he's been doing. Well, maybe not all things, some of the things. He'll highlight what he's been doing for his doctoral research at Oregon State University. And so I encourage you all to join us again Friday noontime for Corbin's second talk. And thank you so much, Corbin. It was wonderful. Really appreciative of what you've done today.
Awesome. Thank you for having me, everyone.
Take care.
Bye, everybody.